Hi students, now let's continue with mutations. In a previous section, we had spoken about the point mutations, and now we are going to discuss about the large mutations which can happen in the genes. Sometimes what happens, copy number variations can happen in the case of genes where it's a type of mutation where large chunks of DNA are inserted, repeated or lost. And these regions of DNA can be around to be 10,000 to 5 lakh bases long. Other than that, you can also have duplication of genes where there would be an increase in the number of copies of a gene. You can also have deletions of large regions in the chromosome. You can also have loss of one copy of a gene, or probably it could be loss of both copies of the same gene. And also you can also have movement of a section of DNA from one location to the other. These all will, rather than being point mutations, these are included under the large mutations of the gene. So let's go on. Coming to the types of mutations, you can, based upon their occurrence, you can classify mutations into two main ways. One is a spontaneous mutation, the other one is an induced mutation. Spontaneous mutations are those which arise occasionally in all the cells, even in the absence of any added agent. And they mainly result from the errors during the DNA replication and also from spontaneously occurring lesions in the DNA, and also by the action of various mobile genetic elements such as the transversons. So three main reasons can contribute to the spontaneous mutations. One is the errors during the DNA replication. The second one is the lesions in the DNA. And the third one is the mobile genetic elements or the jumping genes called the transversons. On the other hand, the induced mutations, they are the result of exposure to a mutagen, which can either be a physical or a chemical mutagen. Different mutagens, we'll be going to the details of that in the next sessions. Okay, so mutagens are some things which will induce a mutation, which will trigger the mutation to happen. Normally it doesn't happen only in the presence of an external agent. It can be a physical agent, like sometimes even UV light can be a mutagen, or it can be a chemical agent. Some of the additives that we add in our food can sometimes serve as mutagens, or you have also heard about mustard gas. Okay, so all these, when the mutations are being induced by something from outside, that is referred to as an induced mutation. Now let's come into the spontaneous mutation and its causes. I am talking about the first cause, which is mainly tautomerism, which could result in a spontaneous mutation. Normally, if we have to talk about what is tautomerism, tautomerism is a situation in which, okay, the nitrogenous bases will they shift from one isomer to another tautomeric form. Okay, if you want to go to the details of that, just look here. The normal form of occurrence of nitrogenous bases, they mainly occur in the keto form. Okay, but sometimes a rare imino and an enol form can happen. Okay, these are the rare imino and the enol forms which have been depicted here. And when they occur in the rare imino and the enol forms, what happens? The base pairing rule will get altered. What is a base pairing rule? The normal base pairing rule is that adenine pairs with always thymine and guanine pairs with the cytosine. So if they occur in the amino form, what happens? That base pairing rule is being observed. But sometimes when it goes into the enol form, what happens? Enol or the imino form, what happens is this base pairing rule will be shifted. So let's see what is it. This is the standard base pairing arrangement. You have here thymine, which is in the keto form, and adenine, which is in the amino form. So adenine and thymine have a hydrogen bonding here. You can see the hydrogen bonding here. Okay, and cytosine and guanine, they are in the amino, and they both are in the amino, and one is in the amino form and the other one is in the keto form. Okay, so what happens is here guanine and cytosine is pairing, and in this case adenine and thymine is a pairing. This is the standard base pairing rule. But what happens is sometimes this thymine can convert itself into a enol form. What is the enol form that you have here? Just look at this. The H of this N is shifted to the keto group here. So it becomes a OH. Instead of having a keto group, what happens? It has been converted into a enol form. You look at this. This is the keto form of the thymine, and what happens? A hydrogen from NH is getting shifted onto that keto group, and it becomes a enol form. Same way, look at here, the cytosine. Cytosine is having a amino form, that is NH2. But if you go to look here, cytosine is the H is being converted into is is transferred to the next N. So it becomes the amino form becomes into a immuno form. In such a conditions, these are mainly the tautomers. Okay, they are not defective bases, but they are tautomers. What are tautomers? Tautomers are isomers of a molecule that exist in a solution or in a cell. And they are interchangeable forms because the chemical bonds are rearranged many times spontaneously. And this is quite different from chirality, where the molecules are mirror images of each other. And the DNA bases, as we have seen here, they will undergo various tautomeric shifts. And what happens is, when the DNA replication is happening, if tautomeric shifts are happening, instead of A and T binding with each other, what happens? Thymine, if it is in the enol form, it will base pair with guanine. Okay, and if cytosine is in the immuno form, that would bind with adenine, and this would lead to what? Mutagenesis. So there will be a mutagenesis if tautomerism occurs during DNA replication, such that the new daughter strand incorporates the incorrect base pair to complement to the tautomer. So this is an example of that. I think it's quite evident. So you have the from the normal form, that is a keto and amino form of base pairs, they get shifted to the enol as well as the imino form, and those tautomers will have a false base pairing, resulting in a change in the base pairs. Now this is a consequence of mutation as a result of tautomerism. What happens? The temporary enolization of guanine leads to the formation of an AT base pair in the mutant, and a GC to AT transition to the mutation occurs. And the process usually requires a 2 replication cycle. Mutation only occurs if the abnormal first GT pair is missed by a repair mechanism, and the wild type is in the form of the gene before mutation occurred. Okay, usually when this happens, when such tautomeric shifts happen, sometimes what happens, you will have, now here you can see here, this is GC. Here it is being paired and it is converted into GT. But here it is GC. So this is a first generation progeny. And what happens here, it is GT. And sometimes these errors can be repaired by certain DNA repair mechanisms. Okay, but if the DNA repair mechanism is not identifying this at the time point, and if this mutation is being undergoing, again, this is the first generation of DNA replication. In the second generation of DNA replication also, if it is being carried out, that is being transferred. Okay, so the mutation becomes more prominent when it is being transferred by the second generation. Okay, so here what happens, the GT is converted into the wild type, and this is a mutant here. You get a mutant here. Instead of G here, the A also is coming. Okay, so a mutant can be available, can be more evident in the second generation. Okay, and this process, if it is being identified by the DNA repair mechanisms, it will not be identified. Now, the spontaneous mutations can also be of 2 different types. Okay, you can also classify them into the transition mutations as well as the transversion mutations. Now, what is a transition mutation? The shift from one form to another changes the hydrogen bonds characteristic of the bases, allowing one purine for another purine or a pyrimidine for another pyrimidine substitutions. That is, when a purine has been replaced by another purine or a pyrimidine has been replaced by another pyrimidine, such a mutation is referred to as a transition mutation. and that could eventually lead to a stable alteration of the nucleotide sequence, and it is a relatively common type of mutation that we have. Then the next type of mutation that we have is a transversion mutation, where a purine is substituted for a pyrimidine and a pyrimidine is being substituted for a purine. And this has been found to be very rare due to the steric problems of pairing purines with purines and pyrimidines with pyrimidines. Okay, so the difference, that is adenine, which are the purines. Okay, if a purine is being replaced by another purine, okay, that is referred to as a transition. But if a purine is being replaced by another pyrimidine, then it is referred to as a transversion. I hope this is clear. And the spontaneous mutations can mainly happen also due to the insertion and deletion of nucleotides. Okay, you can see a different case. These mutations generally occur when there is a short stretch of repeated nucleotides is being added. Okay, in such a location, the pairing of the template and the new strand can be displaced by the distance of the repeated sequence, leading to insertions or deletions in the new strand. Now look at this particular strand. You have a slippage leading to an insertion. Okay, what happens here? GT is being undergone to be, what happens? It has been slipped. And as a result, what happens? Another addition here, T gets added. Okay, and a slippage which is leading to a deletion. So you can have here GC triple T is a 1. Up in the parental strand, what happens? A slippage is happening, and this is the parental strand. Here a slippage is happening, and so what happens? It will result in a deletion. So what we got to say is the causes of spontaneous mutations are mainly tautomeric. It could be some errors during the recombination or during the replication of the DNA. One reason that we had to say is a tautomerism, and the second one is the slippage of the nucleotide bases, which could lead into the insertion as well as the deletion of nucleotides. Hope so this is clear to you. We'll be continuing in the next sessions. Thank you.